Hello everyone. So for this video lesson, we're going to explore about applications of DNA science. But before that, let's have just a simple recap about what is this DNA, how it was developed, and how it affects our lives. So in 2001, a major scientific milestone was announced. Researchers had completed a first draft sequence of all 3 billion base pairs of the human genome, only the fourth eukaryotic genome to be sequenced. This news electrified the scientific community, and few among them would have dared to dream that a mere 9 years later, genome sequencing would be underway for more than 7,000 species. By 2010, the researchers had completed sequencing more than 1,000 bacterial, 80 archaeal, and 100 eukaryotic genomes, with many more in progress. Ultimately, these achievements are attributed to advances in DNA technology, the methods of working with and manipulating DNA that had their roots in the 1970s. A key accomplishment was the invention of techniques for making recombinant DNA or rDNA. These are molecules formed when segments of DNA from two different sources, often different species, are combined in vitro. When we say in vitro, in a test tube. So the advance gave rise to development of powerful technologies and techniques for analyzing genes and gene expression. All right, so in this one, we will going to explore as well how biotechnology affects our lives, how the manipulation of organisms or their components to make useful products. And biotechnology includes such early practices as selective breeding of farm animals and using microorganisms to make wine and cheese. Today, biotechnology also encompasses genetic engineering, the direct manipulation of genes for practical purposes. And genetic engineering has launched a revolution in biotechnology, greatly expanding the scope of its potential applications. Now, tools from the DNA toolbox are now applied in ways that affect everything from agriculture to criminal law to medical research. So, DNA technology is in the news almost every day, right? Most often the topic is a new and promising application in medicine, but this is just one of the numerous fields of benefiting from DNA technology and genetic engineering. So here we're going to explore the DNA science and how it comes a long way, and its applications or technology have brought about so much change in our lives and the environment. So in this video lesson, it therefore aims to present biotechnologies in the perspective of DNA science as a springboard. Specifically, at the end of this lecture, we should be able to identify various biotechnologies as applications of genetic engineering, to analyze the pros and cons of these technologies, and perform several exercises on these technologies in the fields of medicine, agriculture, industry, and forensics. Okay, so let's think about this. How did DNA become a reliable tool for forensic science? Paano nga ba, 'di ba? How it becomes so dependable way or technique to identify kung sino yung criminal, kung sino yung suspect. And how also this kind of technology help us to identify who is the biological parent of a certain individual. Okay, so DNA is now a science word that a lot of people worldwide are somehow quite familiar with. This, however, was not the case before 1985 when DNA analysis was first proposed by Alec J. Jeffries to be used in investigating law enforcement cases. Medyo matagal na din pala itong ginagawa. So let's have a portion of a news clip about this. Alright, so please allow me to read this newspaper in which this shows how genetic test traps girls killer. And this is a killer breakthrough in which the day DNA evidence first nailed a murderer. I think it was in the year 1980s. Okay, so this newspaper was written by Paul Harlan. Okay. A man who killed two 15-year-old girls and was caught as the result of the first mass screening for genetic fingerprints was sent to prison for life. He had tried to avoid the test by taking a workmate into taking it instead. Mr. Justice Otten sentencing coined pitchfork age 27, and at Leicester Crown Court said he was a danger to young women. Had it not been for the development of the genetic fingerprints test, which identifies unique chromosome patterns from a sample of blood or saliva, he might still be at large. Pitchfork of Littlethorpe, near Leicester, who is married with two children, admitted strangling teenagers Linda Mann and Dawn Ashworth in 1983 and 1986, I think. So he was sentenced to life imprisonment on two counts of murder and was also jailed for 10 years on two counts of raping the girls. He admitted to charge of indecently assaulting two other girls and was jailed for 3 years on each count. He also received 3 years for conspiring to pervert the course of justice by putting Ian Kelly to take his place in a genetic test. All sentences are to run concurrently. So Kelly, age 23 of Leicester, admitted conspiracy and was jailed for 2 years, suspended for 18 months. The prosecution accepted that he didn't know he was shielding the killer, so the deception was revealed by other workmates. So the court heard that Pitchfork had killed Lindemann after driving his wife to an evening class. He left his 4-year-old son in a carrier cot in the car while he stalked the girl on a footpath near Navarro. He was alleged to have told police, "I ripped her clothes off and jumped on her and beat her up." Mr. Brian Scott Cox and QC prosecuting said he exhibited great self-control and must have been a total lack of remorse. Neither his wife, whom he killed up later, nor anyone else suspected he had been involved in this terrible crime. 3 years later, he carried out the identical rape and murder of Dawn Ashworth, who suffered appalling injuries. A man working nearby heard her screams but thought someone was playing about. Pitchfork told police he killed the girls because he feared they would identify him. The court heard he enjoyed exposing himself to young women and was a sexual psychotic. So this mentioned case was solved through DNA fingerprinting or DNA profiling, whereby DNA barcodes or profiles generated from biological specimens collected from crime scenes are compared to the victims on suspects' DNA for confirmation as a crime perpetrators. This DNA technology is also utilized for purposes of paternity as well, and confirmation of relationships, as well as providing historical and evolutionary relationships. So, ganyan ka-powerful yung technology, 'di ba? Pwedeng bigyan tayo or bigyan ng hustisya ang isang krimen. And it is really helpful nowadays, 'di ba? Okay, now let us discover how important this polymerase reaction sa atin, o yan, sa society. Okay, so the polymerase chain reaction has enabled scientists to quickly identify segments of DNA, much as your fingerprints may be used to identify you. As we know, each one of us has its own unique fingerprints. This process is called DNA fingerprinting, and it makes use of non-coding sections of DNA that consist of 2 to 5 bases repeated over and over again. So, as in, halimbawa, yung pag halimbawa na-repeat yung DNA sequence natin, halimbawa, ng adenine, thymine, guanine, and cytosine, ito yung mga bases ng DNA natin, diba? So, people differ by how many times such a sequence is repeated. But how can this difference be detected? Paano nga ba? PCR is used to amplify the entire repeated region. The greater the number of repeats, the longer the segment of DNA that will result after PCR amplification is done. People be heterozygous for the number of repeats, as you can see here in the figure. So the DNA from both homologous must be amplified separately. Following PCR, the DNA is subjected to gel electrophoresis. As we know, ano ngayon? This is a process that separates DNA molecules according to their size with an electric current through a jelly-like material. So, the shorter the segment of DNA, the farther it migrates through the gel material in bonding pattern. DNA fingerprinting has many uses, as we know. Medically, it can be, or it can identify the presence of a viral infection or a mutated gene that could predispose someone to cancer. In forensics, very important din yung DNA fingerprinting. From a single sperms, kasi malalaman natin, this is very enough, kahit yung sa strand ng hair natin, to identify the suspected rapist, di ba? Because the DNA is amplified by PCR. And aside from that, it can also be used to identify the parents of a child or identify the remains of someone who has died, such as victims of the natural disasters. Na kaya nga very important itong method na ito. So in the future, we'll be undoubtedly identify the parents of a child or identify the remains of someone who has died, and more applications of this recombinant DNA technology that will greatly enrich our lives and improve our health. So as you can see here in the figure, halimbawa meron tayong na-collect na sample sa crime scene at meron tayong ebidensya, halimbawa itong dugo, and from the blood, makikita natin yung DNA doon, and then halimbawa may dalawa tayong suspect. So yung by performing the PCR, yung repeats, ilang repeats ba ang nakita natin sa PCR during the process, makikita dito, ayan, by using this gel electrophoresis to identify criminals. Ayan, kita natin, ayan, the PCR is used to generate copies of specific regions. As you can see, these variations may be used to identify deceased individuals or suspects in crime, as what I said. So in this example, the evidence suggests that suspect A is not the criminal. Kasi isang repeat lang siya, 'di ba? As compared dito sa suspect B na pareho sila na nakita doon sa crime scene. Okay, that's it. Let's move on. Now, let's have some connections. Ayan, meron tayong question dito. How is paternity test done and why are the results always a percentage instead of a certainty? Bakit nga ba? So, in a paternity test, a sample from the mother or child and possible father are taken and amplified by PCR. Then the samples are electrophorized, and the bonding patterns compared. In a typical paternity test, 13 to 16 human identity markers or the HID are examined. These markers or genes are found in all humans but in different lengths of repeats which are inherited from parent to child. When a certain amount of these markers match in the child and possible father, the result is a percentage of certainty that the man is the father, normally 99% or higher, only by comparing entire genomes, which contains hundreds or millions of base pairs and approximately 25,000 genes na meron tayo, diba? Can a paternity test result in 100% certainty? Okay, that's it. All right, so let's explore more about this DNA fingerprinting. DNA technology, though DNA profiling has just revolutionized the field of forensics, it too has continued to be contribute to the modernization and further enhancement of agriculture, medicine, industry, and many other fields of study. This especially so considering the development and use of GMOs, or the so-called genetically modified organisms, or transgenic organisms. Here in this figure, it shows an example of a glass frog that is endemic in South America. In Japan, Japanese scientists had tried to genetically modify or engineer a native non-translucent frog, which produced F2 offspring. When we say F2, ito yung second generation offspring that has a translucent skin, as you can see here, making its internal organs very visible, allowing easy anatomical and physiological observations without necessitating this dissection. Diba? Hindi na kailangan mag-dissect pa kasi nakikita na dito yung kanyang internal organs. Ganon ka-amaze itong ating DNA technology. Okay, let's move on. Today, bacteria, plants, and animals are genetically engineered to produce biotechnology products. Organisms that had a foreign gene inserted into them are called transgenic organisms. Transgenic bacteria recombinant DNA technology is used to produce transgenic bacteria grown in huge vast, okay, called bioreactors. And nakikita ngayon sa figure na yan, yung mga machine na yan, yung mga malalaking machine na yan. Tawag dyan ay bioreactors. The gene product is collected from the medium, and biotechnology products on the market produced by bacteria include insulin. Alam natin, di ba yan? Yung human insulin, kailangan lang ng vector, yan ba, yung isang vector, usually ito ay E. coli, tapos gagamitin ang kanyang plasmid para makapag-produce ng insulin. Aside from that, the clotting factor 8, human growth hormone, tissue plasminogen activator, and hepatitis B vaccine, and more. So transgenic bacteria had many other uses as well. Some have been produced to promote the health of plants. For example, bacteria that normally live on plants and encourage the formation of ice crystals have been changed from frost plus to frost minus bacteria. As a result, new crops such as frost-resistant strawberries and oranges have been developed. So bacteria can be selected for their ability to degrade a particular substance, and this ability can be enhanced by genetic engineering. So this just show us that hindi lahat ng bacteria ay pathogenic or disadvantageous, parang harmful sa human beings. Meron din namang mga helpful. And for example, naturally occurring bacteria that eat oil can be genetically engineered to do an even better job of cleaning up beaches after oil spills. Ayan. Further, these bacteria are given suicide genes which cause them to self-destruct when the job has been accomplished. At nagagawa na yun ngayon. Ginagamit na yun ngayon. And it was successful indeed. Okay, so GMOs are organisms with artificially altered DNA. The alteration of the DNA in this regard may be through the number one, say for example, the insertion of foreign DNA into host DNA. Aside from that, the alteration of an existing gene. And number three, the deletion or turning off of a gene. Okay? Yun yung mga ways. Okay? So the addition of foreign gene to a host DNA will make the host organism express the trait to, and the host organisms that are made through this process are more aptly called transgenic organisms. The alterations of an existing gene, on the other hand, may cause an expression to be a higher rate or vice versa. And this process is usually used for gene therapy. And we're going to discover that later on. The deletion of a gene is, in other instances, may cause the prevention of the gene or of traits such as the deactivation of the ripening gene in tomatoes. Diba? Yung ating mga ibang mga products na ngayon na siguro hindi natin alam, kinakain na natin. These are transgenic organisms. So let's take a look at this figure. Ayan. Let's have an example at a transgenic mice kung saan kinuha yung kanyang hair. This is used to help burn patients and others by making human facial parts like ears, nose, lips, and others. So, ang ginagawa nila, in-inject nila yung isang gene na kung saan para ma-remove yung hair. At ginamit nila, kasi alam naman natin ang mga daga talaga ay, yung mga mice ay ginagamit na experimental material. So, from it, pwede na ngayong gumawa ng body parts of human beings. Halimbawa, nagkaroon tayo ng aksidente o nagkaroon ng something na or injuries, so pwede na tong gawin actually. So, 'di ba? This is so amazing. But here's the question. Do genetically modified organisms or the GMOs harm health? GMOs are relatively new, right? Naturally, people wonder whether GMO plants and the foods that contain them are safe to eat. Let's explore. So, there are already diverse and far-reaching results with the creation of GMOs, such as the extension of shelf life or of fresh produce, the cell production of pesticides or herbicides by crops and weeds, the improvement of crops such as bigger sizes, natural mechanisms for environmental cleanup, production of desirable chemical products such as vitamins and insulin, and the development of vaccines in a shorter time frame, as well as livestock improvement. Ang daming benefits yan, ng mga GMOs. Okay? Marami ng binibigay sa atin na positive way ito. Very helpful siya. Though there are plenty of benefits and desirable consequences in the use of GMO, still, ayun nga, there are many issues on safety and ethics raised that it becomes a necessity to carefully weigh the risk and advantages of using them in reference to human health, well-being of other organisms, and the environment. Ang dami din kasi talagang nagko-contradict. So let's see. Ayan, let's take the Bt corn as an example in analyzing the pros and cons of using GMOs. The Bt corn is one of the widely propagated crops worldwide, which is an artificial genetic engineering origin. Simply put, these corn varieties are a result of the addition of a gene from the soil bacteria. Ayan, galing sa isang bacteria. Okay? Bacillus thuringiensis ang tawag doon, that allows the natural production of a pesticide in the form of Bt toxin of the corn's genome. In-insert natin ito sa corn, and therefore, Bt corn can produce its own pesticide to ward off pests, such as the corn borer, leaving the corn's kernels intact and in better quality. Alam naman natin itong ating mga corn ay palaging pinipeste, 'di ba? Palagi yang may mga insects na dumadapok kaya nagiging low quality yung nagiging result and product. The patent for this GMO is, however, controlled by very few rich biotechnologies companies, and farmers need to rely on the terminator seeds in fertile soil or provided by these companies. Yung mga gumagawa nito, yung mayaman, 'di ba? Kasi ayun nga, ikaw magtatanim ka, tapos kakainan din naman ng mga peste, meron namang way, kasi itong corn na ito, they can able to produce its own pesticide. Kaya, kahit tumapo man yung peste na yun dyan, hindi na siya mabibigyan ng ganyang sakit, ng mga fungi and others, na it can cause na magiging low na yung kanyang cost as well as yung kanyang quality. So, ayan nga, meron tayong tanong dito. Is Bt Acorn harmful to humans? Ayan, or Bt corn harmful to humans? Typo error lang yung may A. So, let's take a look at this kind of bacteria. Ang tawag doon ay yung Bacillus thuringiensis, di ba? This is a soil bacterium that produces insecticidal toxins. Genes from Bt can be inserted into crop plants to make them capable of producing an insecticidal toxin and therefore resistant to certain pests. And did you know that, according to some researchers, there are no known adverse human health effects associated with Bt corn? Walang harmful effects ang paggamit ng mga transgenic organisms or yung paglagay ng bacteria, itong Bacillus thuringiensis, kanyang genes sa corn. Okay. So let us then try to analyze the consequences of using this genetically modified crop. Ito yung table showing analysis of the pros and cons of the Bt corn utilization. The first column is the field of study concerned. Second is the advantages. Next, the disadvantage and the ethical issues or questions. Let's have in agriculture, yung advantage nito, higher crop yield, 'di ba? Mas maraming naaani. Better crop quality, magaganda yung quality, malalaki, tapos mga malulusog yung mga plants as well as hindi siya walang sakit or walang mga fungi na nakadikit sa kanya. Yung disadvantage naman ito, induces monocropping. Ibig sabihin, isa lang, walang variety, mono, and discourages the planting of several crop species and varieties, as what I said. Wala nang pagkakaiba-iba since pare-pareho yung napoproduce na crop ng mga transgenic organisms. And the ethical issues or concerns about this is that, is it a possibility that pests such as corn borers develop resistance to the Bt toxin produced by Bt corn? Yun yung ethical issue na nag-arise sa waste na ito. Ayan, sa case na ito. Next, let's have economics. Ayan, anong advantage? Since there is more corn produced, the price for corn and its byproducts may be cheaper. Diba? Sobrang magiging mura talaga. While the disadvantage, monopoly control of agriculture by biotech companies. Hindi na nabibili yung mga products na tinatanim ng mga local farmers kasi yung mas prefer na bilihin ng tao ay yung mga magaganda yung quality at naaapektuhan yon yung ibang mga magsasaka, 'di ba? Yung mayaman lang, syempre yung may mga malalaking companies na kayang gumawa nito. Okay. And the ethical issues or question is that, how do farming practices by small-scale farmers get to be affected? Ayun. Yun talaga ang isa sa mga question. Next, we have health. Okay? And the advantage, there will be higher crop yield, more can be fed. Ayun. Yun yung advantage talaga. Since marami ang pwedeng ma-produce, marami ding pwedeng mabigyan at mapakain. Wala itong disadvantage. But the ethical issues for this is that, will human consumers be affected by the Bt corn or toxin in Bt corn food products? Nasagot natin kanina, walang adverse effect according to researches. And how will the consumers know if their food products are produced by using Bt corn? Do consumers need to know if their food products are from GMOs? Totoo naman talaga, right? Bumibili lang tayo sa palengke but we don't know kung yun ba ay kung saan galing yun, ano yung ways na paano yun tumubo or paano yun in-harvest or ano yung quality nun. Kasi wala tayong alam talaga. Unless kailangan nating kailangan tingnan natin, 'di ba? Next, ecology. Ayan. And wala itong advantage, meron siyang disadvantage naman. Monocropping as well causes eventual soil nutrient problems. Syempre, since maraming it can able to stand pests in kung anumang klaseng soil, yung mga nutrients within the soil itself ay nagkakaroon ng depletion. At yun yung disadvantage. Kasi pwede kang magtanim any time of the year, any time of the month, since meron kang isang way para maka-adapt yung plant na yun. And the ethical issues for this: can the Bt corn's genes pass down to other native corn varieties and altogether alter the corn genome through hybrids? At marami na talaga ngayong mga hybrids na mga corn, 'di ba? Meron na nga iba't ibang kulay, iba't ibang laki, and many, many more. So that's it. That's the analysis of the pros and cons of the Bt corn utilization. Moving on, ayan. Other examples of technologies which are offshoots of DNA or DNA science include gene therapy, through gene delivery system that provide correct versions of faulty genes to patients' cells by using various vectors such as lipoproteins and viruses. Gumagamit sila ng mga vectors para siyang host lang na kung saan doon nila ini-inject yung genes, yung desirable genes. Say here, a cell with no functioning gene, ang gagawin, adding DNA containing a functional version of the lost gene, and then it will function again normally. Ganon yung gene therapy. Ganon siya ka-helpful. Ito yung ibang mga technologies na ginagamit ng DNA science. Okay? Next, transgenic organisms used as biofactories for the production of human protein, such as using E. coli to produce human insulin. Oo nga naman, ginagamit ng factories, nagkakaroon ng mga biofactories na kung saan nakakaproduce na ng insulin. And we know, napakalaking tulong ng insulin lalo sa mga diabetic na tao, 'di ba? So, yan. These are the advantage of or ways of the DNA science. And of course, cloning. Ayan, by embryo splitting or nuclear transfer such as that of making a Dolly the sheep and Sissy the cat. Ayan, let's explore kung paano nga ba ito ginawa si Dolly. Ayan. Siya yung genetically identical, first genetically identical mammary or mammal, I mean. Ayan, so yung application nito, this method is used to produce cloned animals whose nuclear genes are identical to those of the animal supplying the nucleus. Ayan, makita natin dito kung paano na-produce si Dolly, the first reported case of mammal cloned using the nucleus of a differentiated cell. Ito, meron tayong dalawang pinagkuhaan ng cell, mammary cell donor at yung egg donor. Sa mammary cell donor, kinultured mo na siya, and then, para siyang ginutom, inarrest yung cell para magbago. Kinuhaan siya ng part during the cell cycle, causing de-differentiation. At yung isa naman, from the egg, yung kinuhaan yung nucleus niya. Okay? Yung egg cell from ovary, kinuhaan niya. Then, fused. Of course, yung nucleus na ginamit dito sa mammary cell donor, ayan. And then, grown in culture. Ayan. Then, syempre, it developed into early embryo and it grows. And then, inimplant ito sa uterus of a third sheep. Ayan. Inimplant, kaya nga tinawag siyang surrogate parent or mother. And then, it develops yung embryo. Ayan. So, nakadevelop tayo ng lump. Ang tawag doon ay si Dolly. The genetic makeup of the cloned animal is identical to that of the animal supplying the nucleus. Sino yung supply ng nucleus? Yung mammary cell donor. But offers from that of the egg donors. Ayan. Differ from that of the egg donor and surrogate mother. Iba yung kanyang genes. Ayan, yung kanyang genetic makeup doon sa egg donor as well as the surrogate mother. Ayan, so if you want to know more about this, pwede nyo siyang makita sa internet. Maraming explanation how this kind of cloned mammal ay ginawa. Alright, so ito namang si Sissy, the first clone cat and her single parent. Yung nakita niyo yung rainbow na nakita niyo on the left side, it donated the nucleus in a cloning procedure that resulted in Sissy Wright. However, the two cats are not identical. Hindi sila magkapareho. Rainbow is a classic calico cat with orange patches on her fur and has a reserved personality, while the CC has a gray and white coat and is more playful. Ayan. Isa lang siya, walang fertilization na nangyari dito. Clinoned lang talaga. At ito yung na-produce, si CC. Ayan. CC kasi from the word itself, cloned cat. Ayan. So much for that. Let's move on. Okay, next is the stem cells and tissue engineering that introduce embryonic or pluripotent undifferentiated cells to damage organs for repair. Halimbawa, meron kang nagkaroon ng sakit or in a certain part of the body or in a tissue, pwede nang gamitin or ang ginagawang ways or techniques doon is yung stem cells and tissue engineering. Let's explore more about this. Ayan, halimbawa ito, yung bone marrow. Halimbawa yung isang tao ay hindi nagpuproduce yung kanyang bone marrow ng red blood cells. Ayan, gagamitan ng gene therapy at using a retroviral vector. Ito yun. In which yung first one, insert yung RNA version of a normal allele into a retrovirus. Gumamit sila ng virus. Tawag doon ay retrovirus. Okay? Kinuhaan yung capsid niya. Ayan. And then, yung meron siyang DNA. Kasi ito mga virus, may single DNA lang mostly. Ito sila. So, then, let retrovirus infect bone marrow cells that have been removed from the patient and cultured. Ayan, kinulture siya. Then, the viral DNA carrying the normal allele insert into a chromosome. And then, inject engineered cells into the patient. Ayan. A retrovirus that has been rendered harmless is useful as a vector in this procedure, which exploits the ability of a retrovirus to insert a DNA transcript of its RNA genome into the chromosomal DNA on its host cell. As you can see in the figure, the foreign gene carried by the retrovector is expressed. The cell and its descendants will possess the gene products. And cells that are produced throughout the life, such as bone marrow cells, as identical candidates for gene therapy. That's it. Let's move on. Next is the xenotransplantation, or the transplantation of organs from one species to another. When we say xenotransplantation, transplantation, is the transplantation of living tissues, cells, and organs from one species to another. While the xenograft, this is an organ transplanted from one species to another. Yung allotransplantation, it refers to same species transplant, while the xenozoonosis is the disease of the transmission. Ayan. Ito yung ginagamit usually at nangyari to, I think, ito, March 9. Ginamit ito. In which, 'di ba, nabalitaan lang natin si David Burnett Sr. Again, he is the man who got the first big heart transplant, but he died after 2 months. Ayan. Kasi itong si just a story about this. Itong si David, ayan, siya ay merong terminal heart disease, who became the first person nga to receive a genetically modified pig heart. Pero namatay siya. According to the statement from the University of Maryland Medical Center in Baltimore, where the transplant was performed. Ayan. So, Bennett received the transplant on January 7 and lived for 2 months. Medyo tumagal din naman siya 2 months after it. So, although not giving the exact cause of his death, hindi pa nila alam kung ano yung ikinamatay talaga, the medical center said Bennett's condition began getting worse several days before his death. Oh, grabe, no? Yung puso ng baboy yung nilagay sa kanya. So, when it became clear that he would not recover, he was given compassionate palliative care and was able to communicate with his family during his final hours. So, nakatulong din itong xenotransplantation sa kanya. Kasi nakausap pa niya yung kanyang family bago siya mamatay, 'di ba? And sa mga nabasa ko, as yun nga, pag halimbawa na ginamitan ng mga ganitong technique, hindi talaga tumatagal. Kahit yung mga clone natin, yung si Dali kanina, hindi siya tumagal din kasi susceptible sila sa mga diseases and other pathogens as well. So, ayun, alam niyo bang nagpasalamat yung um, medical people na gumawa sa kanya or dahil pumayag siya, 'di ba? So, kasi sabi nila, he just proved that he was brave and noble patient who fought all the way to the end. Kaya, ayun, they are very grateful to Mr. Bennett for his unique and historic role. Kasi siya lang talaga ang pinakaunang taong pumayag na mabigyan ng puso ng baboy or pumayag na magkaroon ng xenotransplantation. And he contributed to the vast array of knowledge to the field of xenotransplantation, which is the process of, as what I said, transplanting organs between different species. Ayan. So, yan ang ginamit nila. That's it. So there are techniques that have been developed to insert genes into the eggs of animals. For example, many types of animal eggs have taken up the gene for bovine growth hormone, yung BGH. The procedure has been used to produce larger fish. Nakita nyo naman sa figure. And others, cows, pigs, rabbits, and sheep, gumagamit sila ng BGH hormone. And also, transgenic pigs may be produced to supply transplant organs for humans as a process called xenotransplantation. Moreover, meron pa tayong tinatawag na vaccination. So, vaccine development in just a shorter time frame. At ngayon nga, 'di ba, nagkakaroon tayo ng mga iba't ibang vaccine sa COVID, sa virus na ngayon, na yung pandemic at this time. So, isa yan sa mga product or techniques ng DNA science. And lastly, the genome projects and the Human Genome Project that map out the entire genome of a species to identify all genes that determine all the characters of a species, including genetic diseases. So, napaka helpful ng technique na ito, ng ways na ito, na kung saan through this, you can able to identify kahit palang nasa sinapupulan pa lang or bata pa yung baby, malalaman mo na kung ano yung possible na disease na pwedeng meron siya. At pwede mo siyang maiwasan. Medyo mahal nga lang. At the same time, through using this kind of project, may mapping up the entire genome ng isang species, we can able to know yung mga characters na madedevelop niya as well as the traits. When I say characters, like yung characteristics niya, kanyang physical attributes by, doing this kind of technique. And I hope we are done. Thank you so much for, for listening as well as for your time. And I really appreciate your commitment throughout the lesson. So if you have any questions or clarifications, don't hesitate to ask me. I am always free to send you some explanations and assistance. Thank you very much.